Welcome to Life in Biology. I'm Dr. Joel Graff. We're working through some Mendelian genetics examples. The previous video showed dominant traits and doing some crosses with those. And so if there's something called dominance, there's probably something called incomplete dominance. And then that's what we'll talk about right now is incomplete dominance. And then there's also another variation of Mendelian genetics called codominance. So we'll go through that in the following video. Now, this is an example. Yeah, I grabbed the wrong slide. This is an example of incomplete dominance. And to make matters confusing, we're going to have a capital C here, with a superscript R or a superscript W. And this is incomplete dominance, but we're using the letter C. And then in the next video, when we talk about codominance, instead of a C, it's going to be the letter I with a superscript. So don't think that this letter here is helping you out decide if it's codominance or incomplete dominance. But anyway, this example here is kind of a classic example of Snapdragon flower color. And so here we've got a superscript R for red and a superscript W for white. So we're going to walk through the cross of parental cross, P cross, of two purebred parents. We're going to find out what the F1 progeny of that cross is, and then we're going to take those progeny and cross them. And this is another example of a monohybrid cross. It's just monohybrid in the example of when it's incomplete dominance. And then we're going to look at what the F2 generation would look like. So parental generation, F1's the first generation from that, and F2 is the following generation. If you're wondering about what F means, it's familial. Okay, so let me grab the right slide here, and we've got our parental cross. And so we've got a purebred red Snapdragon flower and a purebred white Snapdragon flower, meaning they're homozygous for the alleles that they have. So if we set up a Punnett Square, we've got the 2x2 configuration here, but really since 100% of the gametes are going to be capital C upper with a superscript R, we could just make it a 1x1 grid. But anyway, you put the two possible gametes from this parent on this axis, the two possible gametes from the other parent on this axis, and then you go ahead and you fill in the what the fertilized organism or cell would be if you combine those two gametes. And so it looks like they're 100% capital C superscript R, capital C superscript W. And where the parents was, this parent was produced red flowers, and this parent produced white flowers. Because it's incomplete dominance, we're going to have 100% are going to be pink. So if you have a heterozygous for color of flower, where it's a capital R and a capital W, red and white make pink. All right, so you take that first generation which were 100% genotype C superscript R, C superscript W, cross it with another of the same thing, and fill out your Punnett Square here. You are going to end up with one square that has homozygous for the R allele, two squares that are heterozygous for R and W, and one square that's going to be homozygous for W. And so our genotype ratio that I just walked through is described here. So I just counted over here and I put a number to it here. Nothing too exciting. And then we have the phenotype, and the capital R is going to be red. So we have one red. The heterozygous at that genotype is going to produce pink, and there were two of those. And the heterozygous that had the W allele are going to be white. So the genotype is a 1:2:1 ratio, and the phenotype is also a 1:2:1 ratio. So that is incomplete dominance, where the trait of two purebreds is some sort of a mix. If you got black and white, you end up with gray. Red and white, you end up with pink, and so on. All right, like and subscribe or not.